So, as we've seen, Mendel managed to do quite well with the limited resources that he had available to him to generate quite a good model of how heredity worked. Now, it might be surprising to you that Mendel developed only a few terms to describe the model that he had worked on. Only the concept of dominant and recessive, and he also made the concept of hybrid precise. Other terms would come later, primarily in the 1900s, when his work was rediscovered, as we've seen in the first lecture. The first concept, or the first word that we need a definition for, is gene. This replaces Mendel's term for hereditary particle. Now, the precise concept of what a gene is something that is a bit fuzzy. It's open to debate. But for the purposes of this course, we can think of it as a section of DNA on a chromosome that is transcribed and often translated, if it's a protein coding gene, for some sort of functional purpose. Alleles, then, are versions of a gene that have different sequences and often produce different phenotypes, as we'll see in a minute. Alleles are two different versions of the same gene. Same sequences, essentially, with small differences, perhaps. Well, although in some cases the sequence differences can be fairly large. But the point is that it's the same base sequence that is modified in some way. Two alleles will also pair at synapsis during meiosis. Meiosis is the cell division that reduces the chromosome number in diploid organisms, and it's the process by which gametes are made in diploids or new haploid progeny are made in haploids. We can tell if two sequences are alleles of each other if they pair up with one another during meiosis. A genotype is the sum total of all alleles that an organism has, and the outward visible appearance of that genotype is called the phenotype. Locus is a term that describes where a gene is on a chromosome, although it's often synonymous with gene. We could say something along the lines of an organism has this particular allele at this locus, although we could also say this particular allele at this gene. They're roughly synonymous, but it refers to the location of a gene on a chromosome. Two other concepts, heterozygous and homozygous. An organism is heterozygous if it has two different alleles of one gene, or has two different alleles at a locus, we could say. And homozygous means that the organism has two absolutely identical alleles at a given gene or a given locus. A monohybrid is an organism that is heterozygous at only one gene or one locus. And the experiments that we've just been talking about, that Mendel did, were experiments on monohybrids, just the one locus that had two alleles present. What this means is all the other loci or genes in the organism are homozygous. So there's only one particular locus that has two alternate alleles. A dihybrid, on the other hand, is an organism that is heterozygous. So there's two loci where there are alternate alleles. And it's also possible to have trihybrids and so on. Higher orders are possible. Some other core genetics terminology and concepts that we need to cover are as follows. Meiosis is a key concept that we need to understand. Both diploids and haploids undergo meiosis. Meiosis is the cell division that is used to reduce chromosome number and also allow for recombination. Now, we'll talk in a little while what recombination is, so we'll just leave that for now. A diploid is an organism that has two copies of every chromosome, so two copies of every gene, two alleles. Humans are diploids. We get one copy of our genetic material from our mother and one copy of each chromosome from our father. So we're diploids. Other organisms are haploids. They only have one copy of each chromosome or one copy of each gene during their normal life. Now, diploids use meiosis to form temporary haploids or gametes, which then go on to fuse to make new diploid progeny, so 2N progeny. Haploids just do this the other way around. They normally live their lives as haploid or N organisms. They fuse together to make a transient diploid, which is 2N, and then that transient diploid then undergoes meiosis to form haploid progeny. Now, the convenient thing about meiosis, whether it's in a haploid or in a diploid, is that the process is exactly the same. By the time you have a myocyte is a cell that's going to undergo meiosis. It doesn't matter if you're talking about a haploid or a diploid, because a diploid has a 2N cell myocyte that will undergo meiosis, and haploids have taken 2N organisms, fused them together to make a temporary or transient diploid, which is 2N, which will then undergo meiosis. So it's actually the same process. A myocyte is a myocyte, and it is always 2N. There are 2 input haploid sets that go into any myocyte. Each input set will have a certain combination of alleles. When a myocyte forms output or N haploid sets, those output sets of alleles or combinations of alleles may or may not match the input set, the 2 input sets that went in. Those that match one of the input sets are said to be non-recombinant, and those that don't match either input set are said to be recombinant. And we'll talk more about that in future lectures. So alleles of genes on different chromosomes recombine through a process called independent assortment of homologous chromosomes. This takes place during meiosis. So recombination here is distinct from the process of independent assortment. So if 2 genes have their loci on separate chromosomes, you can produce new combinations of alleles for those 2 loci, simply dependent on how the chromosomes assort during meiosis. On the other hand, alleles of genes that are physically connected to one another on the same chromosome actually require some sort of physical breakage and rejoining of that chromosome in order for recombination to take place. So that's a physical event called crossing over, which has a genetic consequence of recombination. The thing to keep in mind, and we'll talk about this more in class, is that recombination is a genetic event. Recombination is new combinations of alleles that were not present in the original input haploid sets that went into the myocyte. Now, there's 2 different physical ways that you can recombine things. Recombination for loci in separate chromosomes can be accomplished through independent assortment, and recombination requires physical crossing over for loci that are on the same chromosome. But the point to keep in mind is that there's the physical events, crossing over or independent assortment, and then there's the genetic event which may happen, which is recombination. It's also perfectly possible to have independent assortment that does not produce recombination, and it's possible to have crossing over that does not produce recombination. What matters is if there are new combinations of alleles that are produced. The other thing to keep in mind is that many organisms have what are called sex chromosomes. Probably the easiest example to deal with here is what we use in humans, the XX or XY pair. So XX for females, XY for males. Now, on the X chromosome, there are many, many different loci, many different genes, and the vast majority of them have nothing to do with sex determination. But the fact that they're present on the X chromosome gives them a unique pattern of inheritance because they're along for the ride on the X. So they show a pattern of inheritance that is sex-linked. On the other hand, all other loci, which are not on the X chromosome, say in humans, are said to be autosomal. Autosomal just means not on a sex chromosome. And the inheritance pattern for autosomal loci is the same for both genders, whereas with the XY pair we see this sex-linked inheritance.